hope you're doing well, my lovelies. So, let's check out what I've brought. But careful. It may contain science. Danger science. At least sometimes. This is a Triphyophyllum peltatum, a woody vine known as an African liana from Sierra Leone. Lianas provide climbable terrain for arboreal species in tropical and temperate rainforests, like in old video games where the interactable textures are slightly different. If you're a lemur or a sloth or even an ant or lizard, lianas are an extremely interactable texture for rainforest puzzle platforming. But lianas are also competitive plants. They grow on trees and in fact compete with their local trees for sunlight, water, and nutrients from the soil to a pretty substantial degree. Forests without lianas grow 150% more fruit than forests with them, which indicates just how intense the resource management element is here, even in the super rich biomes that support lianas. Fortunately, for Triphyophyllum peltatum, it has a secondary strat when the resource competition gets too fierce. Carnivory. We talked about carnivorous plants in the very first few weeks of May Contain Science 3 years ago. Back then, we learned that carnivorous plants tend to grow in places with nutrient-poor, sandy, and bog soils. They still need water and sunlight like any other plant, but where trad plants get their nutrients, things like nitrogen, phosphorus, and potassium, familiar fertilizer ingredients from the garden store, they get those from the soil, but carnivorous plants grow traps and digestive enzymes to capture and extract those nutrients from animals. Honestly, it's kind of like eating the cookie dough rather than waiting the 20 minutes for the cookies to bake and cool. Trad plants get those nutrients from biomass that's decomposed into the soil, whereas carnivorous plants just don't wait for the decomposition part. I get that. I get too hungry for dinner at 8 sometimes too. And in places with nutrient-poor soil, you can't necessarily rely on biomass decomposing into the soils near you. There honestly may not be that much biomass, so you gotta catch as catch can. But there is a cost-benefit proposition to carnivory, more than just baked cookies or crispy, and you lose out on that if you eat them all as cookie dough. Carnivorous traps and digestive enzymes are metabolically expensive to maintain, so if you can run a trad plant build that uses roots for its nutrient collection, you do that, not carnivory. That's why it's unusual to see carnivorous plants in biomes like the rainforest. It's so much more efficient to be a trad plant in a biomass-rich environment like rainforest, but it does make some sense for a liana like T. peltatum, which competes so fiercely with trees to get its nutrients. After all, if the competition weren't really serious, we wouldn't see a difference in tree reproductive rates in forests with and without lianas. But those tree stats indicate that this is a matter of survival. And who knows? Perhaps a matter of pride. Happy Pride, T. peltatum. Back when we covered carnivorous plants that first time, we noted that the genes associated with plant carnivory are present in most plants. But for most plants, they exist alongside trad plant root nutrient extraction genes, which were not conserved in most species of carnivorous plants. Violence is the only option for species like the pitcher plant and Venus flytrap, which exist in the same order as T. peltatum are Dioncophilacea. But T. peltatum does have other options. In fact, most of the time it is itself a trad plant. That's right. T. peltatum is capable of turning into a carnivore on demand as the situation requires. That's a huge competitive advantage over typical carnivorous plants. Most of the time, T. peltatum can rely on the more energy-efficient trad plant method of nutrient extraction from the soil. But as this new article in New Phytologist shows, when nutrients get thin on the ground, or thin in the ground, T. peltatum undergoes a rather more menacing magical girl transformation and adopts sticky glandular leaves to trap insects and digestive enzymes to get their delicious nutrients. See, botanists have known about T. peltatum's carnivory on-demand transformation since forever, but what they couldn't prove is what triggers it. T. peltatum is an absolute pain in the ass to cultivate, and cultivation is pretty critical to creating controlled laboratory conditions. Nature, as you may have noticed, is chaotic and full of confounding factors, and lucky for us, this team recently cracked that problem, unlocking a whole bunch of experimental options when studying this plant, including experiments to figure out what soil conditions incite T. peltatum to violence. Our researchers grew T. peltatum in 4 different conditions: normal soil as a control, and soils deficient in nitrogen, in potassium, and in phosphorus. These are all super important life needs things to live ingredients. But only one experimental group became carnivorous: the phosphorus-starved plants. Phosphates are absolutely mission critical to all life on Earth, a component of DNA, RNA, ATP, adenosine triphosphate, and they put the phospho in phospholipids. Now that's not to put disrespect on things like nitrogen. You can have life without DNA, but you can't have life as we know it without nitrogen. And yet, it's phosphorus starvation that triggers T. peltatum to become a murder plant out of a Broadway musical. The authors note that phosphorus availability is low in T. peltatum's natural habitat, and it gets worse in dry seasons, which is indeed the time of year when we see more carnivorous sticky leaf formation in T. peltatum. And while there is a metabolic cost to growing sticky glue for the leaf traps and the digestive enzymes to extract nutrients from trapped insects, T. peltatum isn't the only plant that expends energy to try to cope with phosphorus deficiency. This study's authors contrasted T. peltatum with a more typical plant, Arabidopsis thaliana, aka thailcress, aka our returning hero mouse ear cress, who grew in moon dirt and therefore is more than capable of handling these experimental conditions. Instead of becoming pesticidal in response to phosphorus-deficient soil, mouse ear cress changes its root morphology and architecture. The plant adapts to these conditions by reducing how deep its roots go into the soil and switching over instead to increasing its lateral root length, as well as increasing its root hairs' length and density. Those adaptations say, okay, we know there's not much phosphorus here, so let's see if there's more farther away. And if there's even a little, all these root hairs will grab all we can get. By contrast, phosphorus-starved T. peltatum barely bothered growing roots, unlike its experimental counterparts that all maintained the classic root strat, while phosphorus-starved T. peltatum went all in on carnivory. Yes, changing up your root system, like mouse ear cress, is comparatively less energy expensive than becoming a carnivore. But T. peltatum may have the more reliable strat here, especially in its home biome. For plants like mouse ear cress, if there isn't phosphorus over here, there's no guarantee there's any over there either. But for a plant from a rainforest like T. peltatum, there's a pretty decent probability there will be bugs to catch. So part-time carnivory not only makes sense as a higher margin strat, it makes particular sense in the ecological conditions T. peltatum is in. But both T. peltatum and our trap plant, mouse ear cress, could clearly both sense and adapt to phosphorus-related nutrient conditions. And one of the next questions in this line of inquiry is whether they use the same info-gathering mechanism to do so. If they do use the same mechanism to find out about phosphorus conditions in the soil, the next question is where exactly in the molecular signaling pathway they diverge. Obviously, their pathways split off somewhere, because one of them grows sideways roots and the other grows malicious intent. But now that these researchers can successfully cultivate T. peltatum, we can actually answer that question, and other questions about how T. peltatum's gene expression changes in phosphorus-starved conditions, in order to contrast those genes with both trad plants and full-time carnivores. Still, satisfying our carnivorous plant curiosity is just one advantage to the cultivation breakthrough that enabled this study. T. peltatum is from one of the only 2 plant orders, and it is the only carnivorous plant that produces naphthyl isoquinoline alkaloids. Although some alkaloids regulate plant growth, most functional plant alkaloids play a protective role for the plant in question. Specifically, you're not going to want to eat me role versus animals that, want to eat them. Because the dose makes the poison, these poisonous protective alkaloids have powerful pharmaceutical properties. You may have heard of morphine, another plant alkaloid, as are nicotine and quinine, which Oregon Trail taught me is an anti-malarial drug. And indeed, naphthyl isoquinoline, quin, like quinine, is showing a lot of promise as an anti-malarial, but also as a new drug for leukemia and pancreatic cancers, and possibly even as a new pain relief agent. Being able to cultivate one of the few plants that naturally produces this alkaloid is a big freaking deal, and it goes to show what kills bugs may also make you stronger.